It's just because the plenary lecture took some time, and I was just waiting for more people to get into the room. And well, I welcome you again, and we are delighted to start our session number 19 about the other topics. As you know, I would like to kindly ask you to keep the microphones and cameras off. Only the session coordinator and presenters may leave these devices opened. So we'd like to remind you that the microphones and cameras should remain off, except for the session coordinator and presenters, as I've already said that. And but feel free to ask questions in the chat, and we love to answer your questions and to try to improve our discussion here. Well, our session dynamic is going to be very straightforward. I prepared a very short presentation of all of your work. I summarized it. So if I made any mistakes with your presentations, please don't be mad at me. Today we are going to have like 10 presentations, so I try to do my best to summarize all of them. But first of all, I would like to apologize for the mistake that I made last Tuesday for not presenting the work prepared by Pedro Ribeiro Moradi. However, today we have the opportunity to discuss his work. So I decided to put his work in the first. He's in the first in the presentation line, let's say. And I will project my presentation here for all of you. Just a second. I'm going to share my screen. So these are all the talks that we are going to have today. And I'm going to here are all the talks. As you can see, the first one, the first presenter is Pedro Moradi. And the title of his talk is Effects of Predation by Planktivorous Fish and Microinvertebrates on the Functional Diversity of Zoo Plankton Shallow Subtropical Lakes. Is Pedro here?
Yes, I'm here.
Okay, right, perfect. So, well, the main goal of his work was just try to understand the effect of predators like fish and microinvertebrates would influence the functional abundance of some species of zooplankton. And there are some traits that were used to respond to these questions. He performed some mesocosmic experiments in Uruguay. He used some fish species for that. I don't know if he used 2 species, but you can clarify that afterwards. And also this species of microinvertebrate here that he used as a predator in the Mesocosmic experiment. So here you can see the treatments that we were using. There was a control with no predator, a treatment with fish, a treatment with microinvertebrates, and a treatment with fish and the microinvertebrates. So there were 4 repetitions for each one of the treatments in this experiment. So here are a few results. I'm not going to go in details here, but you can see that fish was very important to decrease the abundance of the traits that were the zooplankton traits that were used in this experiment. And Pedro can talk a little bit about that afterwards. And the main conclusion was that fish had a greater effect on fast swimming, large size and D-type filter feeding zooplankton. Was actually stronger than the predation effect by microinvertebrates in this experiment. Therefore, there is a difference in the selectivity of predation in relation to the functional traits. So that's actually the summary that I made for this work. So I'm going to open the discussion now. So you are free to ask questions to Pedro right now. Does anybody has any question? Let me stop sharing that. So does anybody have any questions for the work of Pedro? I have one actually. Pedro, I was just wondering, what do you think about the treatment fish plus macroinvertebrates in your experiment? Were the macroinvertebrates eaten by the fish? So wouldn't it be better to somehow isolate the microinvertebrates within the treatment with fish? And then you could, I think, have a better view of the response of the prey for fish and macroinvertebrates predation. I know that you had a treatment just with the macroinvertebrates, but I wonder if you put them together and protected the macroinvertebrates that were somehow predating the zooplankton, if this would have some influence in the results of your experiment.
Give me one minute to think.
You can write your answer, Pedro, if you are too shy to answer here on the camera. You can write it here, and then afterwards I can read the answer.
Pedro, I'm here if you need.
Hello, good morning.
Hello.
Sorry for my nickname.
Hello.
Okay, André. It's okay. Okay, Pedro, you can, if you feel like, you can just answer the question now, or I can repeat the question as well if you prefer.
So the fish selected by this work, it's planktivorous fishes, and there was used the two species of the fish in this work, and this in the literature, these species can't eat the macroinvertebrate.
Okay.
She's eats the planktivorous species.
Okay, right. But then what happened in your experiment then? Because when you see the results, the macroinvertebrates are not eating the zooplankton at all. I think you expected that the zooplankton would be eaten by these macroinvertebrate species. So I just wondering why it did not feed on zooplankton. Did it have the capacity to eat or decrease maybe the abundance, the biomass of zooplankton in your mesocosmic experiment? Don't know. Just wondering.
You can repeat the question, please.
It's because when you see the results of your experiment with the treatment only with the macroinvertebrates, there is no effect of macroinvertebrates in the zooplankton. So I was just wondering why the macroinvertebrates has no effect on the abundances of these traits that you selected for your experiment. And then why the macroinvertebrates are not eating the zooplankton? That's actually the question. Why macroinvertebrates not eating? It's not controlling the zooplankton in your experiment.
I will write the question in the chat. Okay.
Rosenberg, thank you for your question. I think comparing the effect, I think the fishes, in all the experiments that we are doing, the fishes almost all the time influence much more than the invertebrates. Yes, probably there was an effect on the invertebrate by the predation of the fish, but I think this kind of macroinvertebrate has no a strong effect.
Median concentration.
Sorry?
Sorry?
I didn't understand.
I think it was just a mistake, I guess.
Yeah.
Otherwise, you can just write it here in the chat, no problem.
Okay. I think Pedro is trying to write. Thank you.
No, you're welcome. I was not actually referring to you, Stella. It's just because somebody spoke something here on the microphone, but it was a mistake. But anyway, so let's move on to the next presentation because we have many in our session. So we have to be a little bit
okay. Yes.
Straightforward. But you can write your answers here, and then after, if you have time at the end, we can just discuss a little bit more. So
I
sorry
think
for
it's
that.
a good exercise for Pedro. He's undergraduate student, so I think
that's great. Actually, I
yes.
really enjoyed his presentation.
Yes.
Was nice.
And thank you for the opportunity to present today.
No, actually, I'm sorry for missing his presentation the last Tuesday. I'm really sorry for that. It was my fault.
No, not at all. Thank you.
No problem.
Okay, let's go to the next one. Let me share here my presentation again. The next one is entitled Cladoceraon Caidorus sphericus feeding on cyanobacteria in lake. I cannot pronounce this name, I'm sorry for that. It's an Estonian lake. And the first author of this work is Ilma Artono. Is Ilma or any of the co-authors here to discuss about this work?
Yes, Helena Gassild is here. I can answer the questions.
Okay, right, perfect. So let's move on. Well, as I told you before, I'm going just to summarize your work. So if I make any mistake, please don't be mad at me. And here there are some very important research questions about this work. How important is cyanobacteria for Caidorus? Which cyanobacteria does Caidorus prefer? Are there seasonal spatial diet changes? Does Caidorus sphericus feed on toxic cyanobacteria? So this is just to contextualize the work. And let's go. So we have some very interesting results here. So it's very interesting to see that the diet of Caidorus was mostly comprised by cyanobacteria, and it did not change throughout time, even in the winter. And here there is a very strong selection for colonial cyanobacteria, according to these results here that we can discuss afterwards. And this selectivity increases towards winter. And it's very interesting results. So some conclusions here: cyanobacteria, both colonial and filamentous, are important food source for Caidorus sphericus in this Estonian lake. This Cladoceraus has a strong preference for colonial cyanobacteria. Most probably this species also ingests toxic cyanobacteria. So the take-home message of these works that Caidorus sphericus can form important link transferring cyanobacterial carbon to the food web in an eutrophic lake. So that's the summary of the work. I'm sorry if I killed your work. It was not my intention, but
everything was correct.
Feel free to ask questions now. Does anybody have questions?
I have one.
Ali, please, Ali, feel free to ask.
Hi, Helen. Thanks for a really interesting study. I never would have think that Caidorus would be one to ingest cyanobacteria, so I learned that, even though you said you have a previous study. But anyways, my question was, if you think Chaidoris is ingesting the planktonic cyanobacteria, or if you think that they're ingesting what sediments after the bloom and what precipitates down to the sediments, especially because of the wintertime signal. So I was wondering if that's like a post-bloom ingestion, or do you think they're ingesting actively also during the bloom? And my second question was, do you have any idea of how the ingestion rates would compare to other grazers like Daphnia or copepods, for example?
Thank you for your question. Hydorus varicus is actually consuming this cyanobacteria throughout its presence in the plankton, and it's consuming the cyanobacteria which are in the plankton. So we have the theory that Hydorus varicus, when it's swimming in the water, it frequently stops on the colonies of colonial cyanobacteria. As we saw from the results that this Gladoceran preferred colonial cyanobacteria. And we think that while it is resting on these colonies, it is simply scrapping the surface cells. The colony of the, for example, microcystis is covered with mucus, and this is full of bacteria, protozoa, and mucus itself is very nutritious. So it might be that it uses this as a very good opportunity to feed, but it also grasps the surface cells. Otherwise, we cannot explain why this zooplankton is specifically feeding on colonial cyanobacteria, especially microcystis, because this cyanobacteria is actually having minority part among the phytoplankton in Wurzel. And what was the second part of the question?
Do you have any idea of the grazing rates, like clearance rate or ingestion rate, in comparison to like how much Daphnia or copepods ingest cyanobacteria? Like, could we say that they ingest at higher rates or no?
Yes, in our previous research, when we actually discovered that this Hydorus is specifically focusing on cyanobacteria, we also had some comparison with Posmina and Daphnia. Posmina and Daphnia, they have different feeding pattern, considering phytoplankton pigments, which we studied. They consumed the phytoplankton pigments in the same composition as they are in the water. But Hydorus is definitely very much different from them. And when Daphnia and Posmina, they generally didn't have a very high proportion of cyanobacteria in their food. It was similar to the water they were living in. But Hydorus specifically is concentrating this material in their food. So even though this cyanobacteria is not abundant in Lake Wurzel, it is also toxic, we know. It seems that this is specifically collecting this type of algae, and it can concentrate this carbon and cells in its body. So it might be that this Hydorus is also a very important vector of toxic cyanobacteria up to food web.
Yeah, very interesting. Can I squeeze in just another question? I'm just
yes, go
curious.
ahead. Go ahead.
Is it possible, it's a more technical question, because I never worked with pigments and I think it's really interesting. So is it possible to measure, like convert that to a grazing rate, like a number of cells ingested per organism per time? Is that possible or not?
Yes, it is. This is one of our aims for the future. We haven't done it yet, but we will be. But actually, we also are using DNA methods. So, for example, we also analyzed the Hydorus gut content for the composition of quantitative PCR. So we can actually finally say how many cells of toxic cyanobacteria or just cyanobacteria or microcystis have been ingesting. So this is possible to quantify the research.
Okay, thank you.
Thank you.
Does anybody have? Dr. Sarma, please feel free to ask.
Okay, so we have been working on the cyanobacteria algal mixture on a variety of Gladocera and other species of zooplankton too. Normally what happens is, in order to prove that the cyanobacteria that you have is toxic, you need to offer 100% cyanobacteria to the Hydorus. then you will know whether or not this cyanobacteria are actually toxic. This is number one. Number two is toxic cyanobacteria in association with alga is not toxic anymore. So we had done some experiments using 20%, 40%, 80%, and 100% cyanobacteria with alga. At certain proportion, cyanobacteria is not toxic, did not become toxic because they use a mixture of both for digestion. The third important factor is the fact that cyanobacteria are present in the gut does not guarantee it is assimilated. Okay, so it requires a population level study, or at least another level of study to see that it is actually assimilated in terms of energy, and the population increases when you feed exclusively on cyanobacteria.
Thank you for the suggestions. Yes, these are interesting options. But we have also thought that we could measure toxicity rate of these hydorus. They are not only accumulating these toxins, but they can also transfer within the intestine system when they are eaten up by fish, for example.
Yeah, I agree with you. That is why it is transferring to the next level. But what happens to its own population?
Yes, that we don't know. That's true.
The second thing is another aspect that I would like to mention is that in Valle de Bravo in Mexico, the cyanobacteria are toxic. This microcystis is toxic to a variety of zooplankton, but not the zooplankton that live there. It means there is some kind of adaptation. So they are Daphnia laevis, for example, that lives with the cyanobacteria in the same water body, is able to utilize even 100% cyanobacteria. So I wonder if you have some kind of adaptation of Chaidoras to the local conditions, including the presence of cyanobacteria.
Yeah, this is definitely a very interesting issue because cyanobacteria is very common species in neotrophic lakes occurring with cyanobacteria. We don't know exactly how is this hydorus adapted to our local lake cyanobacteria, but we know that this cyanobacteria is definitely toxic, the microcystis one, and hydorus is probably transferring the toxic cells up to food web. Whether it is itself infected from these toxins, we don't know yet. It's a good question.
Thank you.
Thank you.
We have time for one last question. Does anybody have a question? Well, if not, thank you very much, Helen, for the discussion. Sorry, let's move on to the next presentation. The next one is entitled How to Enhance the Visibility of One's Publication by Dr. Sarma. Well, I added some important points to be considered here. His work highlights the importance of young researchers to increase the visibility of their work, their own contributions in the academia. The main issue related to that is that there is a trend of delayed recognition of works of an author, because and this definitely may damage his or her career in the academic world and also in the society. So the main goals of Dr. Sarma's presentation was to highlight different approaches using the web without spending a lot of research time, such as avoiding doubtful websites or those that demand financial commitment. For example, predatory scholarly open access journals. I'm pretty sure that everybody here has already received emails with some proposals to publish in this kind of journals as well. That's how I decided to summarize your work, Dr. Sama. If I did something wrong, I'm sorry in advance. So we can start the discussions now. Does anybody have any questions to start our discussion?
I would like to make a comment on this, and
Sure.
I would like to mention actually the reason why it may not be completely relevant to this audience, but definitely it is relevant to the young audience, because they always, many universities, many institutions evaluate, offer promotions based on how many times you are cited, where you are cited, what is your impact factor or H index, among others. So many times you publish an article and you think that you have citations the next day. It is just not possible. Publicity enhances visibility. Visibility enhances citations. So this is a pattern. So the first step for you is, especially the younger people, is to enhance your publicity. But how do we enhance the publicity? Your colleagues don't give publicity to you because they're competitors. Who will give publicity? Users. So how do the users know that you have published? If you have published an article and if that is retained in your brain or in your library and is not reaching the target audience, it will never see light. So it is important that you make your publicity. Just publication in a journal, indexed journal, does not automatically guarantee. There are many hurdles your articles to reach the target audience, because citation profiles undergo a kind of protected system. You will see some senior researchers are always cited repeatedly among themselves. They normally don't take into account of younger publications, publication from younger, less experienced one, because you need maturity. How do you get maturity? You need visibility. And these are some of the steps that I offer. Especially our area of science is not that strong as compared to, for example, recombinant DNA technology. Users are more, the journals are more, the journal impact factors are also very high, and its application is also high. Therefore, basic sciences and limnology, including the shallow lakes, we need visibility among ourselves. This is one aspect I would like to address, and that's why I mentioned this part.
Does anybody have any comment or questions to Dr. Sarman? Go ahead.
Yeah, maybe. Thank you, Dr. Sarma. Just a question. You presented details for a lot of different sites which could be used, but if you have to choose 2, 3, which is your best choice?
Well, depends on, if you want to be visible in a journal for being considered as a reviewer, or if you want to have a simply higher number of citations. Higher number of citations, so Google citations, excellent, but it has a limitation. It has got a lot of unwanted references attributed to you, but it has got high citation. It gives the highest number of the citations of your work, because it can scan diverse databases. So if you, but Google is not considered like ORCID. ORCID is being considered by the journals, because that is only available in their database to consider as a reviewer, for example. Okay, in some countries, Google is not accessible. For example, China, some laboratories, some institutions ban Google. So if your coverage is exclusively by Google, it is not accessible in a few other countries. So you need to have a balance. Universally, the Web of Science, which offers a Scopus, which offers these are the standard databases available regardless of the nationality.
Thank you.
Does anybody have any other comments or questions? I have a personal question, Dr. Sarma. It's, have you ever had an experience with your students or even yourself of this delayed recognition that is driven by the system? I mean, of course, that you have experience to make some comment about these questions. I would like to hear from you.
Yes, I myself had this experience before. I had published one article many years ago, 1995, 1986 from India, and that did not reach the target organs, target people. Then it was cited by John Gilbert and Terry Snell from USA and picked up tremendous force. And finally, it entered the AFA manual. So you need some pull in the beginning. As I mentioned in my presentation, if you are a postdoc person with a well-known person, it is easy to pull because you need to be introduced to science. It happens in any field. For example, if you want to start an automobile industry in Brazil, it's not possible because there are forces that stop you. So how do you enter? You enter a company as a shareholder and know the techniques, tricks, connections, contacts, and finally establish yourself into an independent businessman. The same thing also, it takes a very long time to enter because it is a protected system. I would not like to cite an article of a person that is unknown because we need to know what is his background. Is only one sporadic publication or he has consistency in science, consistently evaluated for his research. Then you have confidence in his contribution. This is one of the important things that I mentioned for youngsters especially. Of course, you are a senior researcher, you don't need it, but it's for younger researchers.
Okay, thank you very much for the advices. Unfortunately, we have to move on now. So let's go to the next presentation. So the next one was presented by Jimenez Santos Marco Antonio, and his talk is entitled as Distribution of Habitat-Specific Rotifer Species from Shallow Lakes in Mexico. Well, his studies goals were to present, his present work aimed to analyze the macroecological features that determine the rotifer diversity in Mexico using nestedness analysis like the mean matrix temperature. And also to characterize if there is a random distribution, high entropy of, and if there is also a structure between the species richness and the habitats, for example, comparing littoral and pelagic zones in lakes, from which different rotifer taxa have been reported. And here just some results of the main rotifer species that have been found in Mexico. And some results, approximately 400 species of rotifers have been reported in more than 100 collection sites from different water bodies in Mexico. And the nestedness analysis showed that we'll call it temperature, but you can correct me afterwards, lower than 10 degrees, indicating that, I mean, it's not the temperature itself, it's just an index that is used to indicate that the species is not randomly distributed when you perform this analysis. But the results also show that it's structured on large scales. The zooplankton communities are structured on large scales as being littoral or pelagic dependent on the trophic state of the water bodies. Well, these were the main results, and we are open for discussion now, and I'm sorry Jimenez if I made any wrong comment about your work. But let's start the discussion. Does anybody have any questions?
Thank you, Rosemary. All you say was fine. Thank you, thank you.
Okay, I actually have one question, Jimenez. I was just wondering, is actually a curiosity. Were there any data from the cenotes, or you did not sample the cenotes? And if so, how do you define it? How do you define the littoral zones from a cenote, for example? I have been to Mexico before, and I visited some cenotes there, and I could not actually visualize the littoral zone from these systems. How would you separate for it? If you sample these nodes, of course. I don't know if you did that in your work.
Yeah, in the works with cenotes, they don't take samples from the littoral zone. They take samples from different depths in the cenotes. So in these cases, we are not talking about the littoral rotifers. When I talk about the littoral zones, is from other lakes or dams where you have maybe macrophytes, or you have a delimitation in the littoral zones. But yes, in deep lakes, the works, the studies is more related to samples from different depths.
Does anybody have any other question? Guillermo? No. Actually, I have another one. I was just wondering if you, I mean, we are talking about rotifers here. It's rotifers, they are generally found basically in every continent in the world. I was just wondering if we could somehow find similar microecological patterns like the ones that you found in your work. Do you think that we can somehow generalize that for other parts of the world, or not really? It's a very Mexican stuff.
Yeah, I think we can find some general patterns. For example, Brachionus are very well know that his distribution is very high. So maybe we can talk about a generalistic, I don't know, maybe feeding behavior or habitat high tolerance. Also, Lecane is now have a high distribution. I think there's different genera well distributed, and maybe we can talk some generalist rotifers. There are some specialists like sessile rotifers that are attached on macrophytes, or there are some benthic species, maybe the Notomatid species, or also some Lecane, and maybe they are more distributed in, for example, rivers or in the sediments from shallow lakes. So maybe we need to look more the species in general to look for this generalist and specialist species.
Okay. I would like to make a comment. Well, in continuation to those who are here, I would like to say that the next year we have International Rotifera meeting in Croatia, and the organizers, one of the organizers, the main organizer is Maria Spolar. She is also participating in shallow lakes now, and then she can provide more information, and then that would be great to compare a world rotiferologist, approximately 100 to 150 strong workers gather there every 3 years, similar to the shallow lakes. That was a tradition started since 1976 and is continued. It is a kind of very close family, the rotifer family. All members interact freely, no parallel sessions, and everybody listens to every talk. So if you are interested, please join us there. We will have better interaction with other colleagues. Thank you.
Okay, thank you for the announcement, Dr. Salman. And does anybody have any other questions to Antonio Jimenez?
Hello.
Hello,
May
Maria.
I just join to that discussion and actually now announcement for Professor Sarma. I don't have the best connection at the moment, but actually I'm also familiar with that actually topic what presented by Antonio. And so I think that littoral and pelagial pattern can be also recognized by actually similar ecosystems, and we can discuss also about that on the Rotifera conference. And I hope that also in my presentation you can see at the end that I announced the Rotifera Symposium in Zagreb in 2022, and we will see how will be managed or online or maybe we can be present and get it all together in live in that the conference. But yes, you are all welcome with contribution and presentation on that conference. So I hope to see you in Zagreb.
Thank you, Maria. I'd love to join, and hopefully we are going to be able to have conferences in person in 2022. Well, I'm going to move on to the next presentation. Thank you very much, Antonio, for
thank you
for your work and also for the discussion. Okay, the next one was presented by Katrit Karus. Are you there, Katrit?
Yes, hi, I'm here.
Okay, I'm sorry if I pronounced your name in the wrong way, but let's go. Her talk is entitled as Comparison of Larval Perch Feeding Between Littoral and Open Water Sites of the lakes. And the purpose of the study was to compare larval perch feeding the littoral and open water sites of Estonian lakes in the spring of 2019. And the authors used some indices of dietary importance in order to evaluate larval and juvenile fish prey importance. Feeding, in order to understand the feeding strategies between these populations of larval and juvenile fish. And so that these are the indices that they used. I'm not going to go into details here because I'm not really the person to be asked about these indices. You can ask the author afterwards. And I'm going to hear just a few results with the indices that I have just mentioned. And this index here is a Hulbert's index, just to try to understand the degree of similarity between resources that were used by these populations in the littoral and the pelagial zone in the lakes. As you can see here, there is a very pronounced difference between the niche breadth between littoral and pelagial zone in these lakes. We can discuss a little bit about that afterwards. And also here a few results of the fructose condition index. It's basically to understand and the healthy of the species, of the fish species that were used here. And you can see that you cannot find very strong differences within each one of the lakes, but we can discuss that afterwards. And well, here the authors made a comparison between the share of ciliates, the percentage of ciliates that were eaten by this perch larvae, and basically comparing the ciliates that were consumed against the ciliates, against the proportion of ciliates in relation to the zooplankton biomass, and I can see a strong relation between these two variables. So this explains that probably perch is not really eating ciliates when the proportion of ciliates is low in these lakes. I'm not going to talk about that, but it's another index as well that we're using to understand the selectivity of the food items that were used by this fish among the lakes that were sampled. And here just a few conclusions. I'm not going to read everything here, but I highlighted a few of the conclusions. When the biomass of some zooplankton species is high, they were actually successfully eaten by perch. And large sized species of zooplankton like Daphnia, and also those species that were equipped with spines, like some species of rotifers, as you can see here, were not eaten by perch, even when they were present in high abundance in the water. And in open water habitats, consumed food items were more diverse because of poor feeding conditions in the littoral, I guess. But we can discuss a little bit about this afterwards. So you have the word catrit. Now you can open your camera and we can discuss a little bit about your work. Does anybody have any questions? And I'm sorry if I did not present well your work. I think Andrea asked it first, Dr. Sarma, and then afterwards you can ask her. Andrea, feel free to ask questions.
Thank you for the talk. It's just a curiosity. Do you have information about the relative abundances of larvae in the littoral and the pelagic? And which is the age of these larvae?
The age of the larvae, we got it in spring 2019, and then they were 0++ species.
In terms of weeks, do you have an idea? How many weeks?
Maybe in Estonia we studied larvae in the middle of June. Maybe then 3 or 4 weeks, I don't know.
Okay.
And in Latvia, in case of one of the Latvian lakes, we started, we have our studies too early. In case of some lakes, we got fish eggs and fish larvae with yolk sacs, but these lakes were excluded from our comparison studies.
Do you have another question, Andrea, or that's it? Okay.
Thank you.
Okay, Dr. Sarma.
I have a question too. Normally, the fish larvae, when they are satiated, before they are satiated, when they are fully hungry, they feed on easily capturable prey. Ciliates, for example, swimming speed is low. But when they are satiated, they do not eat those that type of diet. They eat on more energetically profitable prey. For example, the copepods, which are difficult to chase, but they chase and eat once they are satiated. Do you have any idea about the satiation levels of the fish larva in the field?
Sorry, I didn't understand your question.
Okay. A hunger level of the fish larva determines what type of prey they should eat. Hunger level. For example, if the fish larvae are fully hungry, they eat prey which do not swim fast, because then the first requirement is to meet the hunger. Okay, once they are fully satiated, then they go for prey that are energetically more profitable but difficult to capture, like insect larvae. So I do not know if you understand the questions. My question is, did you estimate hunger level in different?
No,
okay. Thank you.
Thank you.
Does anybody have another question? Okay, I just have one question. I was just wondering why the pelagiozone has a wider niche breadth. I would expect an opposite pattern because the littoral zone is generally more heterogeneous in resources. So could you talk a little bit about that?
Yes, we thought it too initially this way, but we assume that in littoral sites of the lakes, in case of our study lakes, the food is sufficiently abundant there, a lot of different resources. But and then these perch larvae, they can feed what they like. They can search more nutritive food objects, or and therefore, if perch chose selectively these food objects, then this feeding niche is narrow. But in pelagial areas of the lakes, perch larvae mostly prey upon food objects they just met. They have no chance to choose between more delicious food objects or to be too petulant. They have to, just what they meet, because in open water sites the living conditions are poorer, and I think because of that.
Okay. Okay, thank you for your answer. Does anybody have any other questions? Or if not, we can move on for the next to the next presentation. Thank you very much, Katrit,
for
your presentation, also discussion. So let's go. The next one, actually, it's a pleasure to present Martin Sundegaard's work. I had the pleasure to work with Martin in Denmark a few years ago. So Martin, if I make any mistake, I'm very sorry in advance. It's a lot of responsibility to talk about your work. But I mean, so the title of Martin's work is The Effects of Temporary Stratification on Nutrient Cycling Between Sediment and Water in a eutrophic lake. Well, there were some questions about these eutrophic lakes. Maybe shallow lakes are not that shallow. Maybe with more dynamic temperature and oxygen conditions than you expect. Effects, for example, in nutrient cycling. What may be taking place in these lakes? Well, just I did some small contextualization, let's say here, and the goal of Martin's work. And it's basically inspired because temporary stratification may have a number of important effects, including those on the nutrient cycling between sediment and water, a change in redox conditions at the sediment-water interface. So in this study, the authors use frequent measurements and sampling during summer in a lake in Denmark. It's a small lake, 12 hectares lake, Amstrup, Denmark. Amstrup Zoo, like the Danes say. And here, just for you to have an idea of the lake, you can have an idea of the depth, the maximal depth, and the mean depth, 2.3 meters. And the concentration of nutrients, this lake is very eutrophic, and the mean summer temperature, the mean summer concentration of chlorophyll is around 60 micrograms per liter. And there are some macrophytes like this species here. And they used a very interesting monitoring system where they took some data of temperature and oxygen in 4 different depths in the case of the temperature and oxygen at the surface and at the bottom of the lake. And they have some very interesting data sets. And I'm not presenting the results of this work because there were a lot of information. So I'm going to be very straightforward here with the conclusions about the stratification of the lakes. So temporarily stratifying lakes like this Danish lakes, we wonder if they wonder if in other parts of the world, if these temporary stratifying lakes are common. There is a very dynamic mixing regimes in the oxygen and temperature, and this may, of course, have some impacts on the nutrient cycling. So high temperature in the water near the bottom of the lake, as opposed to deep lakes, will increase mineralization, oxygen consumption, and release rates from the sediment. This type of lake is probably the worst of the 2 types, non-stratified or stratified, because short-term stratification and mixing events allow high release rates from the sediment and a good contact between deep and shallow water lakes. And these have some important aspects to, for example, green gas emissions to the atmosphere, but we can discuss a little bit about that afterwards. So it's necessary with frequent samplings or automatic monitoring systems in order to determine the frequency and importance of these mixing events in lakes, because they may shift quite rapidly in these kind of ecosystems. So Martin, you have the word now. I'm very sorry if I destroyed your presentation. It was really nice. Well, does anybody have questions? I think Sabine was the first one to raise the hand, so feel free to talk, to ask whatever you want.
Hi, Martin. Thank you for this talk, and this is more an invitation than a question. We have one of these bad lakes in front of our institute door, and we have these intensive long-term measurements, including weekly phosphorus and you name it, chemistry measurements, and in situ continuous measurement of oxygen, temperature, and so forth. So if you would be interested to use those data for, like, you could start with like whole lake P budgets and the influence of these temporal stratification periods on that would be great.
Thank you, Sabine. Yes, I think these events, as I also mentioned in my presentation, are much more common than we think, because for example, in the Danish monitoring systems, we only monitor maybe once or sometimes twice a month. So you will never be able to catch these short-term events, which may impact a lot of things, not only the nutrients, but also the fish and greenhouse gases and so on. So I think they're very important and quite common in many of the shallow lakes we're actually working in. But yes, we can discuss that later. Thank you.
I think the next one was Annette. Annette, feel free to ask.
Yes. So thank you, Martin, for your very nice talk. I enjoyed it a lot. I have actually two questions. The first one is, do you know what is the temperature gradient? You said in your talk it's really small, but from literature I often read that just 1 degree Celsius difference is already enough to have a stratification. I was wondering, is it also within this range for you, or is it less even?
Yeah, that's correct. That usually we use this if it's more than 1 degree per meter, we say it stratifies. But I think the point here is also that you do not necessarily need a temperature gradient. You just need a calm water, calm weather. And since you have these very eutrophic sediments, they will use the oxygen within a few days. So you may not have a thermocline, but you will have an oxycline. And that will impact a lot of things also. So you just need calm weather, maybe warm weather, and then you can have this development within a few days. Mm-hmm. And when you have the next turnover, it will mix again, and you may sometimes have a temperature gradient also, but you do not need it.
Okay, so because in your presentation you showed some temperature gradients that were, well, I have to say you really need a magnifier to see the gradient, so to say, indeed. But still, I was thinking, if you zoom in, could it be a degree of Celsius, or is it really low?
Yeah, but of course, there will be an impact if you have a thermal stratification, because then you have no mixing. Mm-hmm. And in that case, you will have this low oxygen concentrations in bottom waters, which may then take place very rapidly. But that's not always the only situation. You can also have these mixing events based just on an oxycline. So that's even on top of that, you could say, which makes it very much more common, I think.
So, and then related to this question, do you think we need to adjust our classification system for stratifying lakes in that case?
I think you maybe not. Maybe I think you should include a type that's these temporarily stratifying lakes, and you should also be aware that when you do monitoring, and if you do that just once a month, sometimes you see some strange values between 1 month and the next month, but that could be due to some kind this type of things happening in a lake, and you'll never be able to catch these things because you do not measure that frequently.
Oh, no, I agree. Yeah. Okay, thank you.
Thank you, Annette. I think Andrea now has one question.
Thank you for the talk. I was just wondering if then by stimulating phytoplankton, this could have some effect on macrophytes. What do you think about that? The potential effects of temporal stratification could, at the end, affect macrophytes?
Yeah, in the sense that this will speed up the release of nutrients from the sediments. In that case, you get more nutrients to the phytoplankton. In that case, you could also have more eutrophic waters, because in a deep lake, there will be no mixing and the nutrients will leave the surface waters. And in that case, they will become less rich in nutrients during summertime. But these lakes, which are between the shallow and the deep waters, they will probably enhance the release of nutrients from the sediments, including both ammonia and phosphate. So in that case, they would be the worst of the two, because you'll speed up the process of increased loading from the sediment, which will also impact the phytoplankton, of course.
Thank you.
Before Marianne, I think Jessica raised her hand, but it was on the camera. Jessica, please.
Thank you. Yes. Hi. Thank you for your presentation. It was great. And I would like to ask you, if in a lake such as this, with the issues with the nutrient and tunnel loading and the greenhouse gases, if you have any idea of a possible in-lake mitigation measure that could address these issues, and if yes, what would they be?
Of course, it's not easy to change depth of a lake. Sometimes it can maybe, but I think again we're back to probably the nutrients, because if you ensure that you have low nutrient loading, then you will have a clear lake. In that case, you would also have the macrophytes growing on the bottom, and they will prevent low oxygen conditions. So I think the best way you could try to manipulate those systems without changing the water level would be to decrease the external nutrient loading. As for many other things, that's what we come back to. You should try to reduce the external loading in order to improve the conditions of the lake. Also regarding these things that we can see here.
Thank you.
Now Mariana has a question. Good to see you, Mariana.
Hi, good to see you, Rosenberg, Martin. Thank you very much for the session. Your results are very nice, and together with the results of Meredith Holgersson, I don't remember if you were in that session of climate change.
Yes,
Yes.
Yeah.
All the stratification patterns in ponds, plus your results, it seems that we are reaching the point of deciding that shallow lakes stratify and ponds, shallow lakes and ponds stratify much more than we used to believe. And that may be the result of going more often to lakes and ponds, and also the increase in equipment that allow us to have more detailed information on both the water column and also on the spatial, on the horizontal variation within the lakes. So we now know much more about all this behavior, and therefore I think it's a time for us to shift a little bit this idea that shallow lakes are polymictic, except on summer and so on. And I would like your thoughts about that. And also, together with the increase in technology, we have the effects of warming, right? So also stratification is expected to increase due to climate warming and increase in air temperature. So we might be seeing, so this temporal increase in stratification patterns may reflect that we're studying them better, but also a natural pattern that stratification is getting more stable in time due to warming. So how can we disentangle this? So we're not overestimating the effect of warming because we are better in studying this pattern. Thank you, Martin.
Thank you, Mariana. Good to see you again, or not in person. Yes, I think one approach to this is, the first thing is to be aware of these things. They are happening, not only in ponds, because lake here was 12 hectares and some of the other ones I showed were maybe 20 hectares. So this is not only ponds, it's many of the shallow lakes we usually talk about when we discuss the results. So it's quite common. One way to get closer would also to be install more of these automatic samplers. It's do not need to be very fancy. Just thermistors would say something about at least the thermal climate that you can have in these lakes. And if you did that in a number of lakes, as we also heard other presentations, then I think that would make us more wise about these lake systems. And on top of this, you could probably model this also. We have some good modelers in our lab also, and think if they get enough data and the physical things and morphology of the lakes, they would actually be able to model to a great extent at least the thermocline. The oxygen is a little bit more difficult because you need to go out and clean it once in a while, but that would also be very interesting. And about the warming, of course, with the climate change, these things will be probably even more frequently occurring, depending on also the weather conditions, if you have more windy weathers and so on. But overall, you would expect these things to be happening more frequently in the future also. So even more important than they are now.
Okay. I think we should move on, unfortunately, to the next talk. It was very nice discussion with Martin, and it was really nice to see you as well, Martin. And
you too.
Maybe in the next conference we're going to see each other in person.
Yeah.
So let's move on to the next talk. Just a second. The next talk was given by Andrea. So actually it was not given by Andrea, but it was he is one of the author and he's here. I don't know if Matteo is here.
No,
as
he
well.
cannot be here. I will
Okay.
present for him.
All right. So here, well, the title of their work is The Roles of Aquatic Vegetation and Solar Radiation in Driving Yellow Perch Larvae Distribution in Shadow Lake. So here, just for you to have an idea of where the work was performed, in the St. Pierre Lake. There is a very interesting pattern concerning the yellow perch abundances in this lake. And the abundance of these species has been decreasing quite a lot in the last years. And the potential causes of this decline, probably here you can see from this work published in Science of Total Environment last year, which shows that total suspended solids, turbidity and water level are probably the most important, and the phosphorus are the most important variables driving the changes in the abundances of these fish species in the last few years. Based on this, there were some questions that were raised. What would be the expected effects on perch larvae, for example? Turbidity would affect negatively the abundances of larvae, or UV probably would increase mortality because there would be a positive phototropism. So this would increase the risk for the larvae of perch. And submerged macrophytes would probably have a positive effect on the abundances of this fish larvae because it would increase, decrease the predation risk and also reduce the UV risk within the plants. So could behavior help to cope with these threats that I've just mentioned? So these were one of the questions. So larvae are passive or active players. And Andrea can discuss a little bit about that afterwards. So the main conclusions are that there are no direct evidence of UV effects on larval survival or behavior, but there are some interesting results concerning the movement of this larvae. There is a positive phototropism hypothesis. The positive phototropism hypothesis was rejected. Larvae do not aggregate in UV risk surface layers. And larvae vertical position is a function of submerged macrophytes, visible light irradiance at surface and turbidity. So is it because there is an active habitat choice? Well, Andrea can discuss a little bit about that afterwards. So Andrea, I'm very sorry if I if I missed something or if I made the wrong interpretation of your work.
No, we're very nice actually.
I tried to do my best.
Nice summary, thank you. So if there is any question, I will be pleased to answer.
Does anybody have any questions? No questions? I actually have one. I was thinking about I'm sorry, Sabine. I will just ask, and then afterwards you can ask the question. I was just wondering about the zoo plankton migration as well. I don't know if you sampled zoo plankton, but wouldn't be interesting to track the food items that perch larvae eat, for example, concerning the zoo plankton movement in the water?
Yeah, it's a nice question. We have 11 years of samples of zooplankton, but still don't have the time or money to analyze them. Actually, it is a huge effort to analyze larvae. So we focus on larvae, and yet we also have the data about the community of larvae, fish larvae. But still, it was so time consuming only to focus on one of these species. So this is an excellent question, and we would like to answer it, but at this moment, I don't have the zooplankton data. It is on the list.
Okay, thank you for your answer. Sabine, please.
Thank you, Andrea, for the nice talk. I still remember those nice times at the Lake Saint-Pierre some years ago. And
yeah, hi, Sabine.
How far your results now help to interpret the decrease in the fish biomass during the last decades in that lake?
Actually, only in part. I mean, because we expect to find a negative effect of UV radiation, but we did not. So given the population is very low, we were worried about stochastic event, and in some years, if in some years there was extremely high UV risk, there could be a threat, and it was not. So in a way, our work allowed us to put aside this hypothesis. On the other side, we confirm another time that vegetation is key here. So we found that it is important not only for juveniles and adults, but it's important for the distribution of larvae too, both among station and both within a station. Here you have to recall that we're talking about 1 meter depth. So they are shifting down in water column where the macrophytes are. So macrophytes are key, and macrophytes are disappearing in the lake, at least in the last year, as you saw. And that's because a huge increase in turbidity in the system. So partially yes, partially the answer is in part in the decline of vegetation.
Thank you.
Thank you, Sabine. I hope to see you soon.
Any other questions? Okay, if nobody has any other question, I think we should move on. We are almost finishing our session, but well, we still have 3 presentations. Let's try to move a little bit faster. Okay, the next one, the next talk was given by Jessica Papera. I liked very much this presentation, but I'm completely ignorant on the subject. So I really summarized your presentation, Jessica. I'm very sorry for that, because I mean, I know nothing about this subject, but let's go. Jessica and the authors used a very interesting method to fraction the phosphorus in the sediment. Here you can find all types of phosphorus fraction in the sediment of the lake that they sampled in Brazil. And the main aim of the study was to evaluate the potential phosphorus release from the sediment of an eutrophic shallow lake based on the phosphorus fractions that I have just shown you in each sediment, and on an experiment that measured the phosphorus flux between water and sediment. And so here we have the main conclusions of the work, the main results. The main fraction of phosphorus in Lake Estremoz, it's in Brazil. The lake sediment is refractory phosphorus, which is non-mobile. The main mobile fraction is redox sensitive, so it can be released in case of oxygen depletion. Under oxic conditions, the balance of phosphorus between water and sediment is likely to be the most influenced by temperature, and organic forms are particularly relevant. So you have the word now, Jessica, and I'm very sorry for being super short, let's say, in your presentation, but that's what I could do, given my ignorance on the theme. Right, so does anybody have any questions? Okay, I have one question. And congratulations again on your very nice and interesting talk. I don't know if if everybody watched your video, but it was very didactic, at least for me.
Thank you. I was a bit worried. Sorry to interrupt. I was a bit worried because I chose a different approach. Instead of having the PowerPoint slides, just have myself talking and the visual aids, because I thought that would be more engaging. So, yes.
Okay. Well, but that's a question. Would you mind making some comments about the relevance of your work, for example? I think I missed that. I know it's definitely important to understand the dynamics of these fractions in the sediment, the phosphorus fractions in the sediment, but if you had to sell this work to policymakers, for example, how would you do that? I was really thinking about that.
Well, in this case, this lake specifically, Lake Extremois, is particularly relevant because it is used for domestic supply. So therefore, its quality is very important. And you know that in that region, we have a period that has more precipitation and a drier period with no precipitation. And during that drier period with less external input due to runoff, the internal loading could sustain the eutrophication in the system. And that is why it was important to know whether the phosphorus in that sediment was potentially mobile or not. And now we know that most of it is not, it's a good thing, and that out of what is mobile, the main fraction is redox sensitive. It is not a lot of a problem in Extremois because of the well-mixed water column, and there isn't really oxygen depletion near the bottom at the moment. So at this point, it is just kind of good to know, but it could change because we've been seeing an increase in phosphorus concentration in Lake Extremois. Like in 2014, it was considered oligotrophic, and now it already has total phosphorus concentrations and chlorophyll concentrations that can make it to be considered eutrophic. That is quite quick. So the eutrophication could lead to oxygen depletion and maybe the release of that main mobile fraction from the sediment and create this positive feedback. So, and there is also another thing that is useful to know from the mobile fraction of phosphorus in the sediment. That is, if you want to use mitigation strategies such as flock and sink or flock and lock or something to do the capping of the sediment and avoid the internal loading, you need to perform the fractionation of the phosphorus in the sediment and know how much of it is mobile so that you don't waste product with non-mobile phosphorus.
Okay, thank you very much for your answer as well, Jessica. Just an advice, I think you should add that in your presentation. Because
I mean, yes. No, you're
the next
right.
time.
You are right. And I recently had the exercise of redoing this presentation many times because I had to do it for the shallow lakes, and then I had to do an even shorter one, a 5 minutes one for another occasion. And I tell you, the 5 minutes was better because I edited more and I had to work on hand picking the most important things. No, but I agree with you.
Okay, so thank you very much for the discussion. So let's move on for the next presentation. The next one was given by Maite Colina from Uruguay, and the title is Greenhouse Gases from Subtropical Shallow Lakes, Submerged Microphytes versus Phytoplankton or Low Primary Production. As I told you before, I did a very super summary of everybody's work. So I decided to take this picture because it's probably the most important one for the work's hypothesis. So here you have the role of primary producers in the lakes. And the example here for the dynamics of release of carbon dioxide and methane are for temperate regions. So the authors just wonder what could be expected for subtropics concerning the release of methane and carbon dioxide in eutrophic lakes and lakes that are dominated by submerged microphytes. So we have different systems in subtropical regions, so longer daylight hours all over the year and higher temperatures. So what would be the implications of these traits, these characteristics to the release of these gases? So here you have the aims of the work to assess carbon dioxide and methane fluxes in natural subtropical systems from Uruguay with contrasting configurations or states at the habitat and ecosystem level, and also to contribute to understanding the role of submerged microphytes as possible mitigators of carbon dioxide and methane emissions in fresh waters. So they used three lakes with contrasting conditions. So here you can see the lakes. The first one were dominated by submerged microphytes. The second one were dominated by phytoplankton. And the third one was a turbid one, but with extremely low primary production. So here are just some main conclusions concerning the findings of this work. Primary producers play a key role in the release of carbon dioxide and methane in these shallow lakes. High submerged microphytes biomasses might promote carbon dioxide intake at subtropics, even in the winter. But methane production and their emissions are not significantly lower than in phytoplankton-dominated lakes. As a consequence, total emissions of carbon dioxide are neither lower than in eutrophic and phytoplankton-dominated lakes. So the littoral zones play a major role in carbon processing and may support stronger emissions than pelagic zones. That's the summary that I was able to perform. I hope I did not destroy your work. No. And everybody is free to ask questions now. I think Sabine has the first question. Feel free, Sabine, to ask.
Thank you, Maite. That was really interesting. And I was wondering, you said that the increased methane emissions from your macrophyte-dominated system was because of the quality of the macrophyte material. But do you think that also other factors could play a role? As for instance, there is often a very high oxygen deficiency in the early morning hours because of the high respiration rates in dense macrophyte stands. So do you think that this could also have an effect on these high methane emissions?
Yes, could be. Actually, the place that we in the shallow lake that was dominated by the submerged macrophytes, we found the really important methane emissions in the littoral area, and the littoral area was not just dominated by the submerged macrophytes. It was a more diverse area regarding the macrophytes, and of course, I guess, yes, part of the biomass of the macrophytes is what I said, is there accumulating and being a suitable source for methane production because it's not really consumed by the herbivores and so on. But of course, there will be another process there. We know that in the littoral area, there is also in subtropical lakes a high diversity of consumers also. Of course, all of those processes will be contributing for that. I don't know if I answered your question, sorry.
I think it's also because you have a high surface for biofilm. So it's not about only the diversity, but also the sheer mass of bacteria that get the surface provided to grow on, that you have, like during the dark periods, you have a very high oxygen consumption in those dense stands. And I wonder how far this also contributes.
Yes, could be. You said that maybe important community of bacteria could be in the plants. That is.
Yeah, in the biofilm, yeah. On the surface.
The biofilms that, yes,
increase the surface for the bacteria to grow. Yeah,
it could be. Yes, we do not test it, but of course, it's one possibility. Yes. Thanks for the comment.
Does anybody have any other questions? I have one actually. I was wondering why the submerged macrophytes in subtropical regions, at least in your systems, do not reduce methane emissions compared to eutrophic systems, and what would be the mechanisms that would explain these dynamics in the lakes that you sampled? Do you think that I don't know if there is co-dominance, for example, with floating plants that could be somehow having an influence in the release of methane in these lakes? Well, that's what I wanted to ask. I don't know if I was clear.
Yes, I think I understand. Actually, our lake with macrophytes, submerged macrophytes, it has a really huge biomass of macrophytes all the year. It's really huge. Actually, you cannot go with the boat there. It's in some parts because it's a huge biomass of submerged macrophytes. So I guess that biomass is there. I don't know. It's not being consumed, and of course, it's a lot of organic matter there. But actually, where we found, as I say to Sabine, where we found the higher emissions was in the littoral area. And of course, there we have also some free-floating macrophytes too. But we didn't measure on the top of the macrophytes and between them, the free-floating macrophytes. In that lake, we have some kind of huge islands of free-floating plants, and we measure in the shore islands, but not in the top or in between. So I guess that we are observing is not the effect of anoxic conditions created by the submerged the free-floating macrophytes that are usually proposed as a way that this kind of plant can increase the methane production. I think this because really it's a huge amount of organic matter from the plants that is being accumulated there, and from all the different type of plants, emergent, free-floating, and submerged. But that's what I think that is more
because of that. We also have profiles from the lake, and it's well oxygenated in all over in the entire column, water column. So I really think that this is a huge amount of organic matter there.
Okay, thank you for your answer and also for the nice work. And I really enjoyed it. Unfortunately, we have to move on for the next to the next presentation. Thank you very much, Maite.
Thank you.
So let's move on to the next one. So the last talk of today is entitled as Future Threats to Shallow Lakes and Freshwater Systems, Nanomaterials as a Novel Environmental Stressor by Franca Stabili, I think. As I told you, I will summarize. I'll try to be as brief as possible. So the main aim of Sabine's work, not Sabine, sorry, Franca's work, was to evaluate the fate and effects of different nanoparticles at population, communities, and an ecosystem level using constructed artificial wetlands, as you can see here in the picture. So there were some specific questions for this work. So you have here a pilot experiment in order to understand, for example, when nanoparticles are introduced into a wetland, what would the particles, what would happen with the particles? Do the particles come out? If yes, when, how, chronic or as a pulse? So which concentration? Are the nanoparticles retained in the wetland system? If yes, where? So they wanted to know the fate of these nanoparticles. So that's why they performed the experiments, and also test the experimental setup and methodology. And here a few conclusions about the work. The results showed that some nanoparticles moved through the wetland and came out in the outlet water. They found that other nanoparticles were retained in the sediments and were taken up by organisms, which is very interesting. Overall, the distribution could be explained by the internal hydrodynamics of the artificial wetland. And pre-exposure monitoring of the wetlands is needed to understand the nanoparticles effects. They also are planning to perform some pilot experiments in order to understand that. So this was my summary, Franca. Hope that I was clear. And so we are now open for questions. Does anybody have questions for Franca? Nobody has questions. I have one actually, Franca. First of all, congratulations on this work. I think it was very nice. And I was wondering how you used gold in your experiment. You can comment that afterwards. I never thought that there were people using gold for performing experiments. And what kind of effects do you think that the uptake of these nanoplastics may have, for example, in the zoo plankton and phytoplankton, and what would be the effects of this uptake of these nanoparticles to the top of the food webs?
Okay, hey, thank you. Thank you for the question. When we talk about nanoparticles, it's also very needed to specify which material it is. I'm presenting now results of plastic nanoparticles, but of course depends a lot on the material, what will happen. So regarding plastic, for example, there are lab studies that have shown negative effects on fishes behavior and also some metabolic, the metabolisms of lipids, for example, that those fishes have taken the plastics through the food web. So there have been reported some negative effects through the food web. So nanoparticles can move through the food webs and they can have negative effects at different levels. Also in Daphnia, there are several studies. But the aim of our study was to try to see those effects and also the fate in these more natural systems. Because the problem, and that's why we use gold, is that when we introduce the plastic nanoparticles in the system, nowadays it's not possible to track it with the normal chemistry analysis because of plastic composition. You cannot separate what is plastic and what is something nano but natural, because plastic is mainly carbon and other atoms, so it's not so easy to separate. So that's why we use this gold inside the plastic, and it's very little gold. Of course, it's expensive, but it's very little. And the good thing about gold is that we usually don't find gold in natural systems, not in the wetlands I'm using now. So the good thing is that if I find gold, I know that it's from the nanoplastics I have entered. So I hope I can answer.
Yeah, you did. It was actually very nice. Thank you for your answer. Does anybody have any other answer? Sorry, questions?
I have a comment.
Feel free to do so.
Nice to see you on the screen. Congratulations on your work. I think it's a very promising avenue of research all about these new entities, novel entities, including these nanoparticles, pharmaceutical products. So all these new things that we are releasing to the environment. And that's coming on top of the old problems that we all know. These are new ones, and as Rosenberg was asking, we don't know fully what their effects can be on trophic webs, for instance. But as I said, they come on already fragile ecosystems, already stressed communities. So do you have an idea of what could be will they have synergistic or antagonistic or no additive effects whatsoever to the already existing stressors that we have over aquatic communities?
Thank you for the question. I think it's very interesting. And to be honest, I would really like to be able to explore a little during my PhD, combining with also other stressors in the environment. Because also depends, as I mentioned previously, it depends a lot on which compartment of the ecosystems you are looking for, because most of the studies are focused on plankton species, and the little studies that have been done, for example, considering also benthic systems, they also found changes, but most of them in the long term. So it's also something that we should consider when we try to understand these effects, because as you say, these particles enter to systems that are already fragile. And for example, I don't know if different trophic states of a lake will have different responses. So it's still a lot of work needed, but the interesting thing is that we need to do also more natural experiments, I think, because that is really lacking. It's a lot of work done at labs, and I understand that, but it's also needed to explore more natural situations. So yes, I don't have an answer.
You have a lot to do already. Thank you.
Oh, thank you very much for the discussion, Franca, and congratulations again on your work. Well, now we have to finish our session, but before finishing the session, I would like to ask you, all of you, to open your cameras, and then I can take a screenshot and publish it on the social media. If you don't mind, of course. If you don't want, it's fine as well. Okay, so just think that you are on the beach, enjoying a very nice drink, and then you smile. So thank you very much. So see you. I really hope that you enjoyed the session and also the conference. And it was really nice to see some of you here through the camera. Hopefully we are going to meet each other again in the next Shallow Lakes conference, wherever it will take place. Okay, so good to see you.
Thank you very much.
See you. Bye-bye.
Thank you.
Bye-bye.
Bye.